Good evening, everyone. If you can hear me, can someone just do a thumbs up comment or just comment that the sound is coming through okay? Excellent, thank you. So we might just wait a moment to get started, just so that people can join. This is the first time I've tried this Facebook Live approach, so we're all learning as we go. So I'll just wait another moment and we'll get going. I think that the presentation will take around about half an hour or so, and I can see comments on the screen, so probably the easiest way, if there's something that you want clarified as you go, I'll have a chance to do that. So yeah, we can take it from there. So I've just seen one little ground was at the top of the comments screen, not the bottom, so that's a little learning for me. Okay, folks, well look, what I'll do is make a start on this webinar, which we've decided to have a go at, given that we're all not able to attend our club nights or other things. And so this is a seminar that I've given before with a little bit of updating to a couple of poultry clubs, and it's really designed to be an introduction to genetics in exhibition poultry. You don't need to understand genetics to be a good breeder of exhibition chooks, but I've certainly found that a knowledge of genetics has helped me when I've been trying particularly to create new colours or move some things around in my breeding program. For people who don't know me, obviously my name's Kevin Smith. I've been breeding exhibition poultry for about 35 years or so now. Had a whole range of breeds over the years, and also as my job, I work as a professor of genetics at the University of Melbourne. So I earn a bit of money with genetics and I spend a lot of money playing with genetics in the backyard at home. So just as I say, this is an introduction. Some of you will know a lot of this, and for some it's brand new, so we'll just take it as we go. So genetics is the study of traits and how those traits are passed from a parent to their offspring. And there are many, many obvious variations that are controlled by genes. And some of these things are things we take for granted every day, and we might not know that they're actually under genetic control. Now, none of you have your web cameras on, so you can all try this. Who can roll their tongue? And if you look at that tongue rolling trait, it is actually controlled by genetics. And you either can or you can't. So no matter how much you practice, if you don't have the genetic ability to roll your tongue, then you can't. Obviously, we've got some examples there also from people with the dimple on the cheek, also in the cleft in the chin, and curliness in hair. But I'm sure you're all there at the moment trying to roll your tongues and seeing if you have that ability or not. So there are genetics for important things and also things that we don't actually understand why they are important. And the traits that we're talking about are determined by the genes, and these genes exist on chromosomes. And I want to not get too technical, but I think it's important to give you some of this background because you'll see these words when you read something online, and they sometimes add to the confusion that people might have. So the chromosomes are really strings of genes, And the gene is a segment of DNA that determines the trait. So it codes for the trait that you see. And it's the variation in this genetic basis that causes the variation in different traits as to say whether a bird is blue or whether a bird is black. And just to go a little bit further, the chromosomes usually come in pairs, and so genes come in pairs. And this becomes important because we'll often hear that a bird is split for a trait, and that's because one of the genes on one of the chromosomes is different to the other one. So, and we inherit these chromosomes from our parents. So in humans, there are 46 chromosomes in 23 pairs, and we get one set from dad and one set from mum. So this is the way that the genetics are combined and give us the variation that we see. And so this is just explaining here another set of words that will come in that are important when we understand genetics when we breed exhibition birds. So on this set of chromosomes, as I've said, you'll have two different copies of a gene. And if this could be that one of the copies codes for blue eyes and one codes for brown eyes in this example. And we call these different variants alleles. And so they're the different variants of genes that are responsible for the same trait. In this case, blue eyes or brown eyes, but they could be rose combs and single combs as well. So, and when we're looking at the fact that these individual genes combine with each other, that is how we can understand both the genetics of the individual that we're looking at, but also what came from its parents. So you can imagine in this case, one of the parents had genes for coding for blue eyes, and the other one had genes coding for brown eyes. Just takes a moment for the screen change to occur. So here we are. And the next part, before we start talking about some examples, which I think is really what most people are here to see and understand, is you will hear the terms of dominant and recessive genes. Now, the gene that prevents the other gene from showing its effects, we call the dominant gene. And it's dominant because it takes over the expression, and the gene that does not show, even though it is present, is the recessive gene. And in our convention in genetics, the dominant gene we normally write in the genetic codes that people either love or hate with a capital letter or an uppercase letter, and the recessive gene we normally write with a lowercase letter. And this is just a way of allowing us to understand both what genes are there, but based on the combination between the upper and lower case, we can actually predict what you will see as the effect of those genes in the individual you're looking at. So again, another non-chook example here, but this is an example where the dark skin color is the dominant trait and the white skin color or albino trait is the recessive trait. So here's an example, and another one to play along at home is that if we look at our thumbs, we either have a straight thumb or what is called a hitchhiker's thumb. So you can see the bend there. Now, the straight thumb is dominant to the hitchhiker's thumb. So again, it's important here that for some reason, back in history, this trait is coded with a T rather than S for straight or H for hitchhikers. So the capital H means that you have a straight thumb, and the lower case h refers to the hitchhiker's thumb. So again, if you hold your hand up and look at your thumb, you have either the straight thumb or the hitchhiker's thumb. So as I said, in a lot of cases, genes code for things that we think are very important. In other cases, they code for traits that we don't fully understand the importance of. So just now to move to some examples from the chicken world. And some of the pictures that I've used here, I've taken from various places around the web, and they are familiar to some people who have put them up, but they help, I think, to illustrate the examples. So if both the genes of a pair are the same, we say that that animal is homozygous or purebred. And so that can be that they both have the dominant trait or they both have the recessive trait. If we have one copy of the dominant gene and one copy of the recessive gene or allele, then we say that the animal is heterozygous or hybrid, or split is a term that is used in animal breeding as well. And so there you can see there that the example that we have a capital T and a lowercase t, so that's a heterozygous example. So the pictures here, we've got two modern game cockerels of Ian Simpson's in Victoria. The silver trait with the capital S is dominant over the gold trait, which is the lowercase s. So in essence here, gold is not silver. So that's why it has the lowercase s rather than the G for gold. So those two birds that we're looking at there, that's a nice clean coloured silver duckwing male. So he's homozygous for the dominant silver allele, and the gold male, to see the gold colour, so the black-red male there, he must be homozygous for the recessive allele to be gold. Now, a golden duckwing, in our terms, is when the male has one copy of the silver gene and one copy of the gene, the gold variant. And that's why we see that straw-coloured hackle that we see in gold duckwings, that you've got that combination there. The silver isn't completely dominant over the gold, so we see the yellow hackle that we see in gold duckwing males. The next term that I'll just introduce before getting on to some more examples are the words genotype and phenotype. The genotype is the combination of the genes of an organism, so it's the actual genetic makeup of an animal. So the physical appearance that we see, though, is due more than just the genetics, and so we call the appearance the phenotype. So if you think about this, we've got a set of genes and they interact with a whole range of factors in the environment to code for what you see. So an example might be that you have genes for yellow legs, but if you don't feed anything that's either green or yellow, the legs may appear white. So whilst the animal has the genetics for yellow legs, it might appear to have white legs. In contrast, if it has the genes for white legs, no amount of green or yellow feed will turn that bird into one that has no amount of green or yellow feed will turn that into a bird that has yellow legs, so it has the genetics for white legs. So the environment can affect things, obviously. So another example would be a bird that has the genetics to be large. If it's not fed properly, then it will never get that full expression of the trait. And the final term that is used when we describe genetics is the wild type. And the wild type allele refers to the example that exists in the most natural variant of the animal we talk about. And so for us in poultry breeding, it's the alleles that would exist in the red jungle fowl. So you can see here, this is what we would call the wild type. And when you're reading some of the genetic codes online, the wild type alleles conventionally have a plus symbol next to them just to denote that they are the non-mutant form of the allele. Everything else is due to some mutation over time. So I'll just get on to some examples now of genes that are important and how the genetics of building, with a particular emphasis on colour, building colour in chickens works. And the most basic locus or gene that is important is one we call the E locus, and it really codes for a whole range of different chick down colours, but it's really the basic building block of a whole range of colours. So again, hopefully you can see my pointer going around here. We have the wild type, which is the partridge coloration in a jungle fowl or an Old English game or a brown leghorn, has that wild type partridge colour. But there are a whole range of mutants of this E locus. So one which is, and this is ranked in order of their dominance. So the most dominant allele here is one we call extended black. And the extended black will mask all the colour effects that you see. So an Australorp, for example, is black because it has the extended black basis that builds that. If we step down from that, we'll see then we have what we call the birchen allele, and this is the one that you would see in a birchen Old English game, silver Sussex, or birchen modern games, or all the grey Old English game as well. And then the brown locus, and sometimes these are confusing because the most common place you would probably see the brown locus is actually in some of the Colombian Wyandottes. So it doesn't actually mean it's the colour, it just codes for the way they change the chick down. So you can see here that the typical penguin kind of chick that is an Australorp has the capital E. This is a birchen-based chick. This is a Wheaton chicken, the wild type, a brown chicken here. So you can see here that this is also gold-laced Wyandotte. And then this buttercup allele, which is a very rare one. You'll notice here too that there are actually two versions of Wheaton, a dominant one and a recessive one. Most books you will read will say that the dominant one is the most common, but I know from my own crosses here that at least in Old English game bantams in Australia, we have both dominant and recessive Wheaton options in there. So they look the same. They both are the traditional Wheaton Old English game, but one is a very recessive version. The other is dominant, at least to the wild type, but would be recessive to the black type. So if you understand that order of dominance, you can start to understand why if you make some of these crosses, so if you cross, say, a crow-winged Old English game, so a dark grey bird with almost anything else, you're going to get a dominance of the dark grey type birds, at least in that first cross. So the next gene that I'd like to talk about is, so now that we've got those building blocks of the E locus, that's in essence the canvas that all these other genes work across. So I've already mentioned silver and gold, and they are fundamental issues. So all birds will be either silver or gold, even a blackbird, so an Australorp, is either silver or gold underneath that extended black. So again, these are our two modern games from the previous example when we talked about dominant and recessive. And so you can see here, as I mentioned, silver with the capital S is dominant over gold. So the silver is dominant over the black-red. But as I mentioned, if you have one copy of the silver, one copy of the gold allele, or the not silver, you get what we call the gold duckwing color or phenotype. So you'll get that straw-colored hackle there. The other issue that comes in with silver that is important to know is that it's actually a sex-linked trait. So it exists on the sex chromosome, not on one of the standard chromosomes. So what this means is that in birds, the males have 2 copies of this X chromosome, and the females only have 1. So the female can only have 1 copy of the colour. So when you understand this, this is the basis of making all the sex-linked crosses or auto-sexing colour crosses that you see. So if you take the gold male and mate it over a silver female, obviously this is a male, then all the progeny become gold because they get the gold from their father, and so all the female progeny are gold, and all the male progeny have 1 copy of the silver gene and 1 copy of the gold gene, so they end up being this gold duckwing colour. So that's the basis of that sex-linked crossing that we see. That becomes very important when you're understanding moving those colours around, and also the fact that it doesn't work in reverse, because if the female is gold, then she has the recessive allele, so all of the individuals end up with the silver colour. So this is just an example of that sex linkage using a cartoon here of what's meant to be a Rhode Island Red and a Sussex. And you can see here that this is just the example that I showed, that if you make that crossing in that direction, then you get all silver males and all gold females. So the other aspect of this that comes in is that this works in every way when you cross silver and gold together in this direction. It does not work in the other direction. And it also, the ability to see it at day old depends on the basic building blocks of the E locus. So it's very hard. Anyone who's bred Wheatons will know that the soft, creamy Wheaton chicken, it's hard to see whether that's a silver Wheaton or a gold Wheaton. So what tends to happen is that these 10 crosses tend to work best in some breeds versus others when it comes to sexing the chickens at day old. The next gene that we'll look at is blue, and blue probably should actually be called splash, because in its pure form we get the splashed phenotype that we see here in this Brahma. The amount of splashing that you see varies a lot from breed to breed, and it is somewhat based on the kind of blue that you want to see. So in Leghorns, or in this case with this blue Brahma, where the blue is very light, the splashed form tends to have what this sort of flea-bitten colour was what the old Leghorn breeders here in Australia called it, whereas in some breeds where the blue is very dark, such as an Orpington, and the splashed birds get shown, then the splashed birds are actually much prettier than this kind of effect here. So my splashed Leghorns sometimes look almost white with just the odd dirty feather there. I'm sorry, I don't know why that went backwards, so we'll just scroll it forward again. So how blue works is that it's not completely dominant. So it works to dilute the black. So if you get one copy of the blue gene, then you get the blue phenotype. If you get two copies, then you get the splash. So one of the challenges with blue is that the ability of this gene to reduce the black is influenced by a whole range of other traits or other genes. So that's why you get such a variation in blue. And it also doesn't work as well on every feather. So one of the ways that you can always tell a bluebird from a lavender is that a bluebird will tend to have the odd black feather somewhere, which is a real pain when you're breeding them. So you tend to find that the amount of black feathers and the shading of blue that you'll see will depend on the amount of selection that you've put for all these other genes that are in the background rather than blue itself. But the odd black feather just tends to come with the deal, and you just keep your fingers crossed that it's not a flight feather or a main tail feather. The next gene, and so I'm just using examples here where the birds are basically the same bird with the addition of one gene plus or minus. And so the next example is the barred gene, which is another sex-linked gene, like the silver gold example that we talked about before. And the non-barred type is the wild type, obviously, because jungle fowl isn't barred. But the barred form is dominant over it, so it has the capital Bs. It is also, as I've said, a sex-linked trait, so the female can only have one copy of the gene. So if you wanted to make a sex-linked cross in this way, you would cross a non-barred male with a barred female, and then all the progeny that are female are not barred, and all the male progeny are barred. Now, the other thing that I'd like to say is that barred and cuckoo are actually the same gene. So this example here is some leghorn pullets of minor Nolas. They are actually all full sisters. So you can see in this example here, we have the black. If we add the barring gene here, we have the cuckoo phenotype or color, and there's a blue sister in the background there. So they've all come from the same breeding pen. And you can see in leghorns, one of the reasons that the barring is so, I guess, imprecise in a cuckoo bird is the rate of feather growth. And so that very fast feather growth tends to lead to this irregular barring. So that's one of the reasons why Plymouth Rocks and other breeds tend to often have quite slow feathering, because that tends to be associated with the preciseness of the barring. I can see the comment regarding light and dark barring. Now, in some cases, in most cases, the light barred birds are those that have two copies of the barring gene in the male. And so you get that double expression, and it serves to doubly dilute the amount of black. But that's not the case in Australian Plymouth Rocks. So in Australian Plymouth Rocks, unlike those in the US, both lines actually will breed pure. So originally we used to have the case that it was similar to that, but for some reason, and it's just other genes that interact with the expression of the barring, means that they now breed true. But in most cases, what you'll see is that a light barred bird, or in game terms, sometimes that will be called a mackerel creel, where it's very, very little colour there, is because it has two copies of the barring gene, whereas most exhibition type birds will tend to have one copy of the barring gene so that you see more of the background colour. The next gene that I want to show an example of is the one that's called Columbian restriction or Columbian. And so what it does is that it basically takes a bird that would be otherwise black and restricts the amount of black to the neck, tail and wings in this way that we see here. So a light Sussex isn't actually white. She's a silver bird. And here we have the gold version here in the buff Sussex. And the effect of Columbian varies depending on the background of this E-locus that we talked about at first. So if you've got an extended blackbird, it has no effect. So if you cross light Sussex with an Australorp, you won't get a Columbian Australorp. No matter how hard you try, it won't happen. If you have the Wheaten background, such as in a Sussex, you obviously get a light Sussex female here, but one of the telltale things about this, it's the reason why a light Sussex has white undercolour as opposed to a Wyandotte, where it's on the brown background, and a Columbian Wyandotte has the grey undercolour. So this becomes important when you think about bringing a Columbian trait into other breeds, such as Leghorns, or if you want to work with Columbian Faverolles, which happen to be called ermine, understanding what ground colour you require will influence where you go to look for the best source of the Columbian gene. The next gene that I want to touch on in this introduction was a gene that is called dominant white. There are two types of white, dominant and recessive white. And dominant white sounds like it's going to be the stronger form of white. It actually isn't. So it tends, if it only has one copy, it will inhibit the black, but you have some black flecks coming through. So it's like one copy of dominant white is like a cheap coat of paint. So you cover more or less, but you really need the two copies to get a good version of a white Leghorn. The other thing that happens because of this tendency for the black to come through is that also underneath this white Leghorn with the dominant white, she is also genetically either black, blue or cuckoo. And what we tend to find with exhibition white Leghorns is that they often are cuckoo and they often are blue. And that's because you can imagine if you're putting this white paint over these colours, you're going to achieve a better coverage than trying to paint over the black. And this is one of the reasons that when people talk about crossing a white Leghorn with a black Leghorn and creating blues, they're not actually creating a blue, they're uncovering the blue that is sitting under the white already. She can't be a brown Leghorn underneath, and I'll tell you about the reason she can't be that in a slide or two, but even though she's white, she is also carrying the genes to be either black, blue or cuckoo. And so again, when we play with colours in Leghorns, in the first cross, if we cross her to any of these colours, we'll get that white colour with the odd black or stripy feather coming through. And then after that, when we cross those progeny together, we start to uncover all these birds. So if you want to recreate cuckoo Leghorns, you just go fishing in white Leghorns and you'll find them. And I said that she couldn't be brown underneath, and that's because the colour pile is the effect of dominant white on the wild type or Wheaten bird. So it takes out black, but it leaves the red. And so what this can be confused with a splash pattern bird. So if you think what the example there is in Old English game, if we make a splash red. It's often called a blue pile or a blue sport. But you can also achieve a similar thing with the black feathers coming through by putting the dominant white over the black red. And this is, again, if you think about what I said about dominant white not being the best coat of paint over black, it's one of the reasons that it's so hard to get good, clean coloured piles. And so, you know, in breeds like Jubilee Indian Games where colour's not important, people don't tend to spend a lot of time selecting for it or trying to manage the expression of those black feathers. And it's another reason why a pile modern is so hard to achieve, because the natural tendency is to have some of that black coming through. So the best piles will need to have 2 copies of that there to try and sort of achieve that. I mentioned recessive white. A white Wyandotte is recessive white. It needs both copies to express, so it breaks down quickly if you cross it to another colour, but it does mask both red and black. So even though it's recessive to other traits, it actually is the better form of white in many ways because it covers both red and black, and when you've got 2 copies it doesn't tend to bleed through as much. Obviously you'll still get the odd feather, and anyone who's bred white Wyandottes will know that, but it is a more reliable white in many ways than the dominant white as in Leghorns. The next gene I want to touch on, and I'm just am conscious of time, so I don't intend to talk about every possible gene. I'm just looking at Robert's question. Yes, if you put dominant white over gold lace, you will get a white laced gold type of phenotype. You can also achieve the same thing in terms of it being a splash laced red. So both those options are possible, and you'd need, when you're getting those white laced red Wyandottes, for example, to understand what the genetic basis of the person who bred them were. So the mottled gene is another one that's very important to us. It's a recessive gene, so if you first cross with it, you won't see it, but then it pops up in the progeny. And it's the result of V-shaped ticks to large areas of failed pigmentation. So it's not actually putting pigment on there, it's causing pigment to not be developed. So it's like you're not painting the white on, you're stopping the other colour coming through. And the expression depends, again, a lot on what the basis of the bird is. So the lovely little Ancona here, the speckled Sussex and the Exchequer Leghorn are all mottled birds, but you can see that the expression of the mottling varies, and there was a lot of discussion in the early days whether Exchequer was a different gene. It's also sometimes called hysterical mottling, so you can see that whether it's funny or it's just completely out of control, it's not as controlled. And really the Ancona and the very precise marking on a spangled Old English game are that ticking at its finest. The other thing that comes into play is that the amount with which the colouring fails gets worse as the birds get older, so they tend to get more white, what we would call gayer, and it gets less controlled and less precise as the birds get older. So more and more failing in getting the paint on. So again, one of the things that sometimes frustrates me is that, you know, the people might say that an Exchequer Leghorn is a white bird with black dots. It's actually a black bird with white dots, and they're not very well controlled. And in fact, the less controlled, the better. I'm just looking here at Dawson's comment. I am trying to remember to look. And if a white Wyandotte is crossed with a gold creel, will it produce double-barred creel males? Probably not. It would depend on whether the white Wyandotte actually had barring. In most cases, they don't. Sometimes they will, and they can, but there's not the same amount of barring in them because the recessive white works better to cover over whatever the basic colour is. So a white Wyandotte could actually be a gold laced Wyandotte underneath, purely from a colour perspective. The next gene, and I know this is a complicated table, but I want to touch on it, and it's really important, and it's one of the ones that causes things to go really wrong if you don't understand it and you don't understand how it works. And it's a gene we call the pattern gene, PG. It's a dominant gene, and it's what causes lacing, spangling, not spangling as an Old English game, which I've just said is mottling, and penciling. And so an Andalusian is a blue bird with the addition of the pattern gene to cause its lacing. But its expression really depends on the E base. So I talked about the E locus before. So here we've got brown and here we've got birchen. And it also interacts with other genes, and so there's a couple here. This one we call melanotic, which is about putting black. It interacts with Columbian that we talked about before, and it interacts with a gene we call dark brown. So you get these set of colours, and it's not so obvious here, but the partridge Wyandotte, this Indian game here, and Indian games can actually be built on wheaten, so that's one of the things that things are not often always as they seem, are built on this brown base. And then there's a whole range of colours that are built on a birchen base. So this would be a campine, this is a gold spangled Hamburg, and this is a gold seabright. So you can see how adding one gene will take from a birchen bird. If we add pattern gene and dark brown, we get this autosomal barring, which, as I said, is a campine. It's also a red quill or a dark ginger Old English game. And here we've added this melanotic gene, and that turns from this striping into spangling. If we add Columbian, it turns the spangling into lacing. So you can see that it's a really complicated way to assemble these genes and enable you to change from one thing to another. And it also shows that if you mix them up and they're not all in the pure form, how you can get lacing that has holes in it, or spangles that look like they're half laced, or really all those weird looking versions of that. The other thing that's important with this gene is that it has other effects in different parts of the body. So the reason a gold spangled Hamburg has a black tail is that it's built on the brown base, whereas the silver spangled Hamburg with a spotted tail is built on the birchen base. And so that difference between those colours is really essential, and why when people mix those colours we start to see the spangled tail in gold spangled Hamburgs that we shouldn't have. The interesting thing is that the spangles are nicer on a birchen base. So the pure gold spangled Hamburgs never have as nice a spangle as the silvers even when people really concentrate on them. So it's just the way those genes interact with each other. But understanding how these genes work and how you move from one to another explains why when people start crossing white Wyandottes with laced Wyandottes and don't understand the genetics that are sitting underneath the white and don't understand how to keep all these things pure, it's so damned hard to resurrect where you were. So you've really got to go into this with your eyes open and thinking about where to go. So I understand it's very complicated, but it is complicated, and the reason that breeders find it hard is it's also genetically complicated. So just to sort of wrap up a bit, I just want to touch on the fact that the dominance and single and recessive means that we see some standard segregation patterns in progeny, and so these ratios become important if you're thinking about how to go about breeding a new colour or trying to cross out a trait. So here we have some Anconas that David Simons might know, and we're looking here at some rose-comb birds, and this is an example of if you took a rose-comb Ancona and crossed it to a single comb, if they both were pure to start with, all the offspring would be rose-combed in that first generation, but they would all be split or heterozygous for the rose-comb trait. If we mate them together, we expect that we would see 3 rose-combs to 1 single comb. But unfortunately, 2 of the rose-combs, let's say this one and this one, are impure again. And so these heterozygous birds would throw single combs in future generations. So that's the reason why, if you've got single combs in your Wyandottes, and we have a rose comb Wyandot, the genetics are exactly the same, that they keep popping up, because if in a random crossing situation, you're always going to have twice as many of these heterozygous or impure birds as the pure ones. So without doing some back crossing and progeny testing, you just keep having those single combs pop up. So it is very hard to get rid of a recessive trait in the single comb sense in Wyandottes if you don't do the test mating. Just try and move, whoops. I don't know what's happened there, folks. We'll just go back to here. And so the other ratio, and I just want to put this here, and I know that everyone who hasn't done biology to sort of year 12 level will look at this and go, oh, holy hell. But it just shows that when you're moving to more than one gene being involved, we start to see ratios that become really hard. So in this dihybrid cross, which is looking at trying to, if you were trying to get this recessive comb here, the single comb, out of this, only one of the 16 progeny would express the both recessive traits. So you can imagine then, if you're trying to remove those recessive traits, then you've got a whole heap of animals that are carrying them. If you're trying to breed for them, you've got to hatch 16 to get 1 on average. So it really does mean that if you're trying to have a more complex project, you've got to be willing to hatch large numbers. So of course there's more, and I know that people want to talk about leg colour, and perhaps if people are keen, we could schedule another catch up at another time. But the leg colour, skin colour, and feather colour, there are more genes. We find more variants of genes, they're still being discovered. So the variants of dominant white, there are ones called smoky, one called dun, that there are other versions of it still being discovered as we go through and look at crosses with new breeds of fowl, particularly some of the Asian fowl. And we're starting to get a greater understanding of how genes influence each other. So to know that, well, the basic thing is this is gold and this is silver, but how deep is the gold and how deep is the silver is affected by other things. So we really don't know everything despite all this time. But what I wanted to do was to try and not go on forever, because I know I'm either straight after dinner or between you and dinner and the isolation wine or gin and tonic. But I did want to just touch on a few examples and some of the terms that you might see and provide an introduction to, I think, some knowledge of genetics can help us all as we breed exhibition poultry. So I guess if anyone's got any comments, I'm happy to hang online for a while and just read the comments. Unfortunately, you will have to type them. That's one of the Facebook Live gremlins, really, but it's better in some ways than everyone having to sign up. So I'll just watch if anyone's got any questions or comments. Otherwise, we can all get off and enjoy the rest of our Sunday evening. Excellent. All right. Well, look, I can see that there are a couple of, so Robert's asking about frizzles and other things. So look, I might work at putting some extra slides together for some of the other traits. I'm just looking, yeah. So lemon cuckoo, in most cases, is just the cuckoo on a red background or a buff background. Sometimes the lightness on buff rather than red, you'll get the lemon cuckoo because it's putting the stripe, cutting out the buff, and also dilutes the buff a bit as well. So if you think of a black cuckoo, the black bars are the ones that appear blue, and then you have the white bars as well. So if you can imagine, if you put the barring on a red bird, you go to buff and white. If you put the barring on a buff bird, you go to lemon and white. Just looking, scrolling now. Yes, Neville, waterfowl. Some of the genes behave similarly, but there's a whole lot of new ones. So it's there. There are some excellent books on waterfowl genetics, but the same sort of principles apply, and understanding how they affect things such as neck rings and the base they're on does also help when you're a judge and an exhibitor. Nick, white rose combs, the type is not great. Yep. So yes, I'd just keep back crossing to black or blue. It would depend, and you'd know best yourself whether you have dominant white or recessive white in there. I suspect they're probably dominant white, in which case I would tend to cross them back. If you had typey blues or typey cuckoos, I would use those. I'm just scrolling through. Yep. So Andrew, the basis is how the Colombian gene works on those different backgrounds. So the Sussex have a wheaten background, and so it's able to dilute that undercolour much more than it is on the Wyandottes, which tend to be on a brown background. So that's one of the reasons too that in speckled Sussex, the undercolour will depend. They can either be brown or they are often sometimes wheaten as well. So it's a bit of a tell as to how the Sussex were developed. In the old days, they used to be bred from brown Sussex and had the grey undercolour, but more and more in Australia now, we've relied on crossing light Sussex into them to type, and they don't tend to have the grey undercolour anymore. If I want to create rose-comb black Orpingtons from white rose-comb Orpingtons, where would I start? Cross them. And I believe that the white rose-comb Orpingtons would probably be recessive white. So in the first generation, you would get black progeny with rose combs, but they would be impure. In the second generation, you would get 1/16 of the birds to be pure breeding white rose combs. Light leg is recessive to dark leg. So that is the way. There's also a sex linkage involved there. How come you get black and blue splash out of blue Andalusian? It's the number of copies of the blue gene. So the blue Andalusian has one copy of the blue gene, one copy of the black. So you cross them together, you expect on average to get 1/4 black, half blue, and 1/4 splash. Yep. So with silkies, you can get Colombian in them. It's just a bit harder to see the effect on that silked feather background. So, yep. So pearl eye in Indians is its own colour, but there's a whole gradation of eye colour there between red and pearl. So if you've got really dark red eyes, then no amount of changing feeding will do that. Yeah, blue splash and splash are really just the same thing, just the amount of splash that comes through. Yeah, so the red hue on adult birds is common in a lot of breeds. So, and it can also be a hormonal effect there. So, but the Meg, I'm just reading your question. Probably though, that red there, if you're meaning on the front scales, it's probably just a bit of the red horn, and it's just the white, which is the recessive trait, not being able to pull all the colour out of those birds. Australorp, so what goes wrong from black over to black to get 100% blue? So Daryl, that shouldn't be possible. The most likely reason for that is that the blue in Australorps and Orpingtons can actually look very black and be very dark. So it shouldn't happen. Just scrolling through. No, Meg, I didn't cover eye colour. That would be for another day. Graham, there are some books. One that I like that's not too scientific but accurate is one that Grant Brereton in the UK has written. It's called something like Genetics of Wyandotte Colours, but the same genetics apply, and he uses some very clear examples similar to mine of just putting pictures of birds. Yep. So leg color, cuckoo interacts with leg color to clean up the amount of black on them as a rule, but it won't change white to yellow. Lucas, if with your piles, it depends. Most likely crossing pile to pile, if they are purebred, you will just get piles. You might also get some browns depending what's there. Yep. So Jack, just looking at that, with the silver duckwing or grey from golden male to a silver female, the reason is that you, as well as the straw neck colour, there are also tend to be other genes that influence on the amount of collar on the shoulders and other things that mean that it's really hard to maintain that good silver colour, not just on the neck, but also on the wings and shoulders. Lucas, yeah, if you cross a brown with the piles, you'll get browns and piles. That's a very common way of going about things. Guy, if I want to start breeding black Rhode Island Bantams, where would I start? Yeah, to get a yellow-legged black, I would go to a black Wyandotte. Having bred black leghorns forever, yellow-legged black is a pain in the backside, but if you want to have decent leg colour, I would start with a yellow-legged black breed. I wouldn't go to Australorps or others because you'll spend all your time cleaning up the legs. Genetic basis of lavender, it's a different gene, it's a recessive gene. So it dilutes the other colours, works better on black than red, but works on both. And you need 2 copies. So I'm just skimming through. It's good to see some people throwing in their answers to the questions. That's great. So, yep, David, that's correct. The Punnett square is the same regarding that. That's good. So, brassiness on silvers is 2 things. One is just having the gold versus silver story, but there's also the presence of red, which you can get away with, and red shoulders in partridges or black reds. So Andrew, if you look at some of the Dutch Bantam literature, they actually have red-shouldered silver, but it's that red that hangs around that's part of the problem. So look, thanks folks. I'm going to let people go home, but I'm not certain. I don't know whether the live video hangs around or not. As I said, it's the first time I've done this, so we'll see if it stays there. And look, if we want to have another go and think about some more traits, I'm happy to try and put some slides together. It won't happen straight away, but I'll just wait and see what people's feedback is as to whether it was worthwhile, and we'll see how we go from there. So thanks everyone. Hope that everyone who sat through it got something from it. So bye now.